Did anybody go trick-or-treating last night? I live up in University Hills and got all the little ones like 6:30, 7, and then 8 and 10-year-olds until about 8, and then I got all these UCI students coming around looking for free candy. I thought one of them was a student in this class, but she denied it. So. Anyway, we had 4 different kinds of candy, and nobody wanted the Whoppers. So if you came in late and you want one of these, you can have them, come down after class, pick up a little treat. And if you haven't picked up your exam, you can pick it up on the right side. If you have a regrade request, you put it in the box on the left. So just to start with an announcement, you've probably noticed from your syllabus that we're not going to have class on Friday, November 22nd. I don't know if Professor Walsh mentioned this, but the 11th annual Immunology Fair is held that day. Actually starts the Thursday afternoon with student presentations, but all Friday morning is lectures from distinguished immunologists listed here, and then there's poster session with our graduate students and postdocs. And it's something that both Professor Walsh and I need to attend, and so we can't really have class or even an exam because the TAs are all attending as well, which is one reason we don't have a second midterm in this class. So I was looking for Ryan. I haven't seen him. Maybe he would want to have a review session. Oh, there he is. Ryan, do you want to have a review session in class on Friday, the 22nd of November? Think about it. It's tentative, and it's well before the final, but since we're not using the room for class, if people want to show up and he's willing, we can give you an update about that later. But all the TAs are going to be at the Immunology Fair. And the other reason I bring it up is because this is open to you as well. The registration deadline is today, but it's free. If you have any time that morning, you want to come hear a famous immunologist talk about their work, or come by at lunch or the afternoon, get a free lunch and see some posters, you're welcome to do that. Last year, 4 or 5 students in this class did register and went to the Immunology Fair, or at least part of it. And so you're more than welcome to do that. So I thought I would announce that here. Well, now we get to talk about what B cells do and how they get activated, which is kind of fun for me because we've been just talking about the pieces and the CD molecules and how antibodies are formed and their structure. And now we get to actually see how B cells play in the sandbox with the other cells. And then this is my last lecture, and then I'll hand over the rest to Professor Walsh, and you'll learn more interesting immunology as we go along. B cells and the antibodies they produce are considered part of humoral immunity, and that's, I think, the title of the chapter 9 is humoral immunity. It's out of order because normally you would talk about T cell mediated immunity in chapter 8 first, but there's nothing in this chapter that you can't understand without having seen chapter 8 first. But what does humoral immunity really mean? It comes from the old phrase or term humors, the body humors, the fluids like blood and other body fluids. And it refers to the B cell antibody response, and also it's influenced by helper T cells. And this is important for the protection from extracellular bacteria and their toxins, and from viruses to keep them from going from one cell to the next. Before we get into the actual figures, I want to make some definitions, starting with a protein antigen. It's just what it sounds. It's a foreign protein that induces production of specific antibodies but by the host. The protein can be directly recognized by the B cells through its BCR, and peptides from the protein can be indirectly recognized by T cells after those proteins are processed and presented into peptides and displayed associated with MHC molecules on antigen-presenting cells or any cell that has class 1. Now there's another type of antigen we'll talk about, a polysaccharide antigen, which is a repeating sugar structure on the surface of a microorganism. So here's a bacterial cell, here's an antigen. This could be a protein or it could be a polysaccharide, some sort of sugar on the surface. And that can induce immune response as well because antibodies can really recognize any foreign molecule. But because the polysaccharide is not a peptide, does not have peptide characteristics, when the bacteria is broken down inside the the B cell, you won't see polysaccharides binding to MHC molecules and being presented to T cells. But as we'll see, that doesn't mean that you can't make antibodies to those. T cell help is a general term that refers to signals that T cells provide to B cells to help them make the antigen-specific antibodies. In the lectures from Professor Walsh, you'll learn about specific T helper subsets that are specialized to help B cells as compared to other T helper subsets. Okay, so those are the definitions. Now we can talk about the B cell. Its BCR is, as you know, the B cell receptor, same as surface Ig. Shown here is IgM. And there are two components, two functional components, the antigen binding component from the variable regions of heavy and light chains, and then the invariant component, which in the surface Ig, its role is to interact with Ig alpha and beta. It's not interacting with Fc receptors or complement, but it comes to the surface with Ig alpha and beta, and that is responsible for signaling. So only the secreted form of Ig as antibody does this constant region have those other functions. The other important point about this slide is it shows that signals are generated only when multiple BCRs are clustered together at one pole of the B cell that's contacting a surface of a pathogen or some other multivalent antigen. If you just have individual B cell receptors bound to individual antigens in 3 dimensions that are not clustering the B cell receptor at one pole of the cell, you do not get a strong activation signal. And because most pathogens, whether they're bacteria or viruses, do have multiple repeating copies of the same antigens on the surface, whether they're proteins or polysaccharides or other types of polymers, you do tend to get clustering of B cell receptors at one pole of the B cell. So next is figure 9.2, which talks about the activation of naive B cells. And it shows in this example 2 BCRs being brought together. Here are the Ig alpha and betas. And the first thing to know about the cytoplasmic tail of the Ig alpha and beta is they have these little sequences that are shown as yellow little bars within the cytoplasmic tail that have a name, and they're found in many other types of signaling proteins in the immune cells, T cells, as well as Fc receptors on macrophages and so forth. And these are called ITAM motifs, which stands for intracellular tyrosine activation motif. And then when they're phosphorylated in this picture, that's what the little red dots refer to. It's when the tyrosine in the ITAM motif, intracellular tyrosine activation motif, when it's phosphorylated, they designate that, or they illustrate that with a little red dot here. So not just any tyrosine can be phosphorylated. It has to be in a specific sequence context known as an ITAM motif, and we won't talk about the specific features of that, but just to know that it's a tyrosine in a specific amino acid context becomes phosphorylated when B cell receptors are clustered together. Now, how does that happen? The tyrosines in the ITAM motifs are recognized and phosphorylated by a family of tyrosine kinases. These are proteins that phosphorylate other proteins. And that family is known as the SRC family. So SRC, SRC, first identified in sarcomas. That's where the name comes from. And there are 3 members of the SRC family that are found in B cells that they show here. I'm not going to ask you to memorize those, just to know the general class of tyrosine kinases. They're called SRC family tyrosine kinases. And when you talk about T cells, one of these will reappear, FIN, and then there's another one, LCK, that you'll learn about. They're all members of the SRC family of tyrosine kinases. And among many functions they have in cells, a very important one is to phosphorylate these ITAM motifs. In B cells, that would be the ITAM motifs in Ig alpha and beta. That provides a docking site or a signal for recruitment of another type of tyrosine kinase, which is known as SYK, SYK. SYK has specific domains, illustrated here by this little backwards E shape, that can interact with those phosphorylated ITAMs. And then when SYK kinases are brought together, their kinase domains, which are shown down here, can become phosphorylated themselves, and then they will continue to signal, as we say, downstream to other events in the cell, including changes in gene expression that allow the B cell to begin to proliferate and differentiate. Okay, any questions about that? Well, that's just the start, and that alone is not enough to give a strong B-cell activation signal in general. You need a co-receptor. We've learned about co-receptors in T cells, CD4 and CD8. They strengthen adhesion, they amplify signaling. B cells have their own co-receptor, and it's a complex of 3 different proteins. And these are called CD19, complement receptor 2 or CR2. The figure just shows CR2, I added this in. And then CD81. I really only want you to remember these 2. This is the component that is involved in recognizing the complement-coded antigen, and this is the signaling component. So if you see when you put it together in figure 9.5, together with the B-cell receptor, here's a bacterial cell, here's the epitope recognized by the BCR. During the immune response, the bacterial cell is going to be coated with complement, and some of those C3 fragments will be degraded to further smaller pieces, and one of them is known as C3d. That is a ligand for complement receptor 2. And some of these will be close to the antigen, and that will bring CR2 in very close proximity to the B-cell receptor. And along with it comes CD19, which, as you see, has this long cytoplasmic tail, and it also has sites that can be phosphorylated, in this case by SYK, and that amplifies the activation signal. So you're going to see a very similar case for CD4 and CD8. When a co-receptor is engaged, it brings in new signaling components that lead to amplification of that signal. But it's the same signal, it's just augmented or elevated. Still, the B cell isn't fully activated until it gets a second signal. This is just enhancing the first signal. You need a second signal that's provided by T cells in order to get full B cell activation in response to protein antigens. So antigens for which T cell help is required for antibody production are known as thymus-dependent antigens, or TD antigens, because in animals or humans who do not have a thymus and therefore don't have T cells, you cannot make good antibody responses, and these are protein antigens. We'll return to protein antigens and how T cells cooperate with B cells to induce antibody responses a bit later in the lecture. But right now we're going to talk about 2 types of antigens that are thymus-independent, or TI antigens. These antigens can induce an antibody response even when there are no T cells around, for example, when there's no thymus. And that's because these antigens have interesting characteristics that allow them to activate B cells. So let's start with the TI1 antigens. In general, let me just say that T-independent antigens occur because many microorganisms have certain repeating structural features on their surface that can induce B-cell activation. And the TI1 antigens, that structural feature is a TLR ligand. So the example in the left panel and the center panel is LPS, we know as a ligand for TLR4 over here. And on the right, the ligand is bacterial DNA, which binds to TLR9 in endosomes. So essentially, the TI1 antigens provide the second signal themselves, overcoming the requirement for T cell help by engaging a TLR that produces this second signal identified by the extra arrow over here or here. Okay, so how does this actually happen? It's a little confusing, and I sort of wish they didn't include this part over here, but since it's here, let's talk about it. This is actually the more rare case. You can see a bacteria here. It has various antigens on its surface as these kind of pink squares, but it also has LPS, this long extended molecule with a red tip. And in this particular B cell, which is very infrequent as any B cell would be for a particular antigen, the B cell receptor is actually binding to LPS itself. But then other LPS that's on the surface or falling off this bacteria can bind to the TLR4 with these other accessory molecules we learned about in chapter 2. And that leads to a second signal to allow this B cell to be activated, proliferate, and differentiate into an antibody-secreting plasma cell. Now, the antibodies produced by this B cell will be specific for LPS itself. The much more common occurrence is a B cell will recognize some other epitope on the surface of this pathogen. Just one is shown here, but there are going to be 10 or 20, 50 different potential antigens on the surface of any bacteria. But the bacteria will also have LPS, which will activate TLR4, and that will give the second signal for differentiation. Now, the antibodies produced by this B cell will not be specific for LPS. They'll be specific for whatever the antigen was that was detected by the B cell receptor. But the key second signal was provided by LPS that's part of the same pathogen. And a variant of this is a bacteria. It may have LPS, but it also has these, the bacterial DNA. So when the bacteria is taken up by the B cell recognizing this other antigen and degraded, the bacterial DNA activates TLR9 in the endosome, and that provides a second signal to produce antibodies, again, to this other antigen here. So in general, the common case is a TI1 antigen is some sort of pathogen that delivers a second signal through a PAMP binding to a TLR. And in rare cases, the B cell is actually producing antibodies to that same pathogen-associated molecular pattern. Does that make sense? This is often a confusing figure, so I recommend you go back and have a look at it in the text too. Yeah. LBP is LPS binding protein. We may have skipped over it in an earlier lecture, but LPS is a very hydrophobic molecule, and this is basically protects it in the fluid, in the hydrophilic liquid. Now the TI2 type antigens, they don't provide. There's no other like TLR ligand on the surface of the bacteria, but the distinguishing feature is that there's so many repeating copies of the epitope that you get massive cross-linking of B cell receptors. Shown here are 3 or 6. But typically, it might be hundreds of B cell receptors all brought together in close proximity, and that leads to a really strong signal 1, a very strong primary signal that sort of overcomes the need for a second independent signal. And an example would be a bacteria with a polysaccharide coat that's the surface is entirely covered with the same repeating polymer. Now, this isn't in your book, and it isn't in your notes, and it's not even published yet, but I went to a conference last week, or a seminar, where I learned that there are other cells in the spleen that are required for B cells to respond to T-independent antigens. So they may be independent of T cells, but B cells still need help from other accessory cells, and maybe in the next few years we'll learn more about what those are, and they'll appear in a later version of the textbook. But for now, you just need to know there's 2 types of T-independent antigens that can activate B cells to produce antibodies even without T cell help. And the last thing you need to know about this TI responses is that they're usually more rapid than the T-dependent responses. They generally produce IgM antibodies, and they're not associated with somatic hypermutation. So very little class switching, very little somatic hypermutation. So the affinity doesn't change all that much over time. And generally, they don't produce a lot of memory B cells either. Nevertheless, these sorts of responses are useful early in infection before the T cells start to proliferate and begin to interact with B cells to generate these high affinity B cells or memory B cells. Okay, any questions about TI antigens? Well, now we're going to talk the rest of the time about T-dependent antigens and B cell T cell cooperation. As we have touched on earlier, the interaction of T cells and B cells occurs in secondary lymphoid tissues, the spleen, or I should say the white pulp of the spleen, then the lymph nodes, and all the mucosal lymphoid tissues. We're going to go back to chapter 6, where I didn't have time to finish that chapter, and talk about a bit about B cell trafficking, how they get where they need to go in the absence as well as the presence of antigen. We'll start with figure 621. The B cells are shown here as these larger yellow double circles. And then the smaller yellow dots here is a chemokine known as CCL21. So a lot of the B cells in your body are just migrating around between your heart and then into the arteries and then the veins. They come back. But if they're going to get into a lymph node, the way they do that is they're attracted to these little capillaries known as high endothelial venules, HEV. They're attracted there by CCL21 chemokine, which is detected by a chemokine receptor on the surface. And the same exact thing happens with T cells. And that also causes them to migrate across the capillary walls in a process very similar to what we learned about for neutrophil extravasation, again driven by chemokines. That was CXCL8, here at CCL21. And there's another chemokine here, CCL19. These are very abundant in the T cell zone. And T cells are attracted there as well as B cells. But B cells have an additional chemokine receptor that makes them even more attracted to a chemokine known as CXCL13, which is abundant in the follicles, which, as you remember from the structure of lymph nodes, this is where the B cell area. So they get in the T cell area by being attracted to CCL21, but then they keep going to the B cell area because they're brought there by CXCL13 binding to a chemokine receptor that B cells have, but resting T cells don't. Now, we're not going to talk about this. We're skipping over that, so forget about that. And then we'll talk about, go back one to figure 620. So T cells and B cells come in here through the HEV. T cells stay here. B cells migrate to the B cell area. If no antigen is encountered, all of them will eventually go out through the medulla, or the region known as the medullary cords, and then leave through the efferent lymphatic vessel. And then they may go on to another lymph node, or they may go back into the bloodstream. Remember, they came in through the blood vessel, and then they migrated through the HEV here. What about if antigen is encountered? Now, I'm not going to talk much about this slide here, because we're going to go into details. But you can see a lot's happening. T cells are dividing, B cells are dividing, germinal centers are forming, plasma cells are being produced, antibodies being produced. So we're going to talk about all those steps as we go along. But first, I think it's important to tell you, how does the antigen get to a place where B cells can see the antigen? This is something that's really not covered in your textbook, and there's been a lot of progress in the 3 or 4 years since the textbook was published. So some of it, here's antigen. I'm going to denote it by a light blue pentagon here. If you have an infection in the tissue nearby, and this is a draining lymph node, most of the pathogens you hope are taken care of by the innate immune system, but some will survive and go in through the afferent lymph into the capsule area here. Some might be breakdown products, little bits and pieces of the pathogen. So the antigens enter through the afferent lymph. Some of them will diffuse into the B cell zone, but most of them are too big to actually diffuse like that. And what happens is that there's a specialized class of macrophages, abbreviated by the capital M and then the Greek symbol phi here, so macrophage, that line the edge of the follicles and the capsule sinuses here of the lymph node. And they trap these antigens, and they actually transport them across into the B cell area as intact antigens. They don't take them up and process them and present them with class 2. They just trap the entire antigen. There's also classical dendritic cells, which I've shown here by these this polygon here in orange, that their main job, as we know, is to process and present peptides with class 1 and class 2 to T cells, but they're also able to trap intact antigens on their surface. And B cells that have entered the T cell zone through the HEV, if they happen to encounter an antigen presented by a dendritic cell, that will be where they get activated. So some B cells will encounter free antigen here, some will encounter antigen on the surface of macrophages, some on the surface of dendritic cells. Okay, now we'll go back to chapter 9. I've told you that T cells, in this case shown here in blue, as well as B cells, enter the lymph node through the HEV. They can also enter through the afferent lymph. If they've started surveying another lymph node and didn't find anything, now they go to the next lymph node. And when T cells are looking for antigen, if they encounter a dendritic cell presenting peptides, that will induce them to proliferate and begin to differentiate. Now, the B cells, in this example, it's showing them being activated by antigen. It's not showing the dendritic cell But that would generally be the way that a B cell would be activated if it gets activated in the T cell zone. But whether it gets activated here or back in the follicles by antigen on these macrophages, what happens is that they don't, they end up migrating to the border of the T cell zone and the B cell follicles. And later on, T cells migrate there as well, and that's where they meet up. Okay, so the B cell, if it encounters an antigen, it will endocytose the antigen complex with the B cell receptor. And as we saw in the antigen presentation lectures, it will process that and it will present peptides on its surface with MHC class II. And that will allow them to interact with T cells at this area at the near the border of the T cell zone and the follicles. And if they encounter the antigen here in the follicles, they migrate towards that border. I think I just repeated myself, but I'm looking at my notes now to make sure I got everything. So here's a little bit of what I just said. B cells have the antibodies surface Ig here, recognize antigen. That will be internalized into the B cell, processed and presented as peptides with MHC class II. Now, if you have an antigen-specific helper T cell, it can form a stable contact with that B cell through its T cell receptor recognizing the MHC peptide composite surface and CD4 interacting with MHC class II. This stable contact is known as a cognate interaction, and you'll come back to that term in the T cell lectures. The cognate interaction accomplishes two things. One, CD40 is a very important molecule on B cells that interacts with an aptly named CD40 ligand, CD40L, on helper T cells. CD40L is not expressed on resting T cells, but as T cells get activated, CD40L is induced. So when a helper T cell recognizes a B cell, the ligand for CD40 can induce an activation signal to complete B cell activation. But the second part of the helper signal is secretion of cytokines. They're not named here, but one of the most important is IL-4, interleukin 4. So CD40, ligand CD40, and then secretion of cytokines, and here's a receptor for the cytokine, and the most important example is IL-4 and the IL-4 receptor. Now in figure 910, they give you some real images about what this really looks like. And it shows you that there's kind of like a tight synapse that forms between T cells and B cells. A helper T cell adheres to the B cell and begins to synthesize cytokines and CD40 ligand. Here's CD40 ligand, here's CD40. Here and here. LFA1 and ICAM1, our old friends from neutrophils, are also involved in forming a stable adhesion here. And you can see here's the T cell, here's the B cell. And the T cell rearranges or reorients its entire organelle structure and cytoskeleton so that everything is pointing towards the B cell that it's trying to help. And you can see that when you stain for the cytoskeletal protein talin, and immunofluorescence shows that it's all concentrated at this region where the T cell is contacting the B cell and forming this, what's known as the immunological synapse. And then the secretory apparatus also orients in this direction. So all of the cytokines, for example, IL-4 here, if you stain for it, it's all being delivered at this tight little contact here. And that makes the delivery very efficient. Also makes sure that other B cells that are not antigen specific in the area do not get exposed to the IL-4 so they don't get activated unintentionally. So this is all very tightly controlled by the synapse that allows the cognate T cell to deliver the help only to the B cell that's asking for it. Any questions so far? I think that it's a good place to tell you, a lot of people have been asking, how can I study better for this course if you didn't do well in the midterm? In the second part of this course, it's a lot of process. There are going to be some memorization. You got to know CD40L and CD40 and IL-4. But I think you'll do better if you think about processes. If you sit down and ask yourself at the end of this lecture or in a week or two, do I can I really state from beginning to end what happens from B cell encountering antigen to B cell secreting antibodies? Where is the antigen encountered? Where does the B cell go? How does it get there? What's a T cell? Which T cells help it? Where do they come from? What are the molecules involved? So if you think at the process level and then fill in the gaps, first of all, you'll be sure that you understand the concepts. And secondly, you'll realize what actual gaps you have in the specific knowledge of the molecules involved. If you just go back to the notes and say, Fruman says it's all in notes, and you just memorize all those things and not think about the big picture, you're going to have trouble, especially in the second half of the course. It's a lot more process, not just T and B cell cooperation, but T and macrophage, and all the things that happen in inflammation and autoimmunity and transplantation. So try to make flowcharts and diagrams and then fill in the gaps. And then if you realize you're weak on the details, you can go back and try to memorize those again and then repeat the process. Okay, so since we're thinking conceptually, some of you may be thinking, well, you said at the beginning of immunology that the frequency of clones specific for an antigen is very rare. Maybe I said 1 in 10,000, 1 in 100,000. So how do you solve the problem that only 1 in 10,000 T cells is specific for a particular antigen, and the same is true for B cells? If they were all just moving around in the blood, they would probably never find each other. So part of it is that you have these lymph nodes that have an organized structure to allow cells to interact, but it's also because of this process where the T cells and the B cells both change their location so they meet up right at the border of the T cell and B cell zone if they're activated, while the other ones continue to wander around in their separate neighborhoods. Now I want to show you a couple of movies. This will be our break for today, but these are really fascinating, and they're a little old now, but the cool thing about them is they were generated here at UC Irvine. In the School of Medicine, Dr. Michael Cahalan was a pioneer, along with Ian Parker in neurobiology, of using a specialized microscope known as the two-photon microscope to visualize live lymphocytes within live lymph nodes, either kept alive on a dish under the microscope, or even with anesthetized mice living with the lymph node under the microscope with a special surgery. And what we learned, the first thing we learned is that lymph nodes don't look like this with a bunch of round cells just kind of sitting there. So what we learned this is sped up, obviously, but the B cells are in red and the T cells are in green. And this is looking at the different parts of the lymph node. This would be the follicles. This would be the T cell zone. You can see they're not round. They're kind of oddly shaped, and they're moving around. This is in the absence of antigen. They're really just wandering around their zones looking for antigen. They're very motile, and that's their job. They're just looking around to see if they just sat in one place, they have a very low chance of finding a particular antigen if it was rare. And that was one of the videos that was published in the first paper in Science in 2002. Now, the second thing that we learned was a few years later that T cells and B cells actually have this little dance that they do, which Mike Cahalan calls the T cell B cell waltz, with the B cell leading in red and the T cell following in green. And these are images shown at the border of the T and B cell zones after about a day or two after antigen recognition. And all these B and T cells are specific for a particular experimental antigen. And you can see that they form tight contacts, and the T cell follows the B cell wherever it's going. And if you were to have the ability to stain this conjugate here, you would see talin here, probably IL-4 being secreted towards that B cell. But you can see this really doesn't look like two little round boring cells. This is a dynamic process that was first visualized here at UC Irvine. And I think it also helps to think about immunology that way, that T cells and B cells and every other cell is constantly on the move, and that they form dynamic interactions. So try to enhance your thinking about immunology by thinking in 3 dimensions and 4 dimensions, including time. So you can go to his website if you want to see more of these videos. This sort of technology is now standard around the world, and we've learned a lot about how immunology really works as opposed to in a 96-well Petri dish. Okay, so we've covered how B cells find antigen in the first place. We know that they meet up with T cells at the border of the T cell zone and the follicles, and we know what happens when they meet up, how the T cell helps the B cell. What are the next steps that happen? Okay, in figure 913, on the left, it shows one possible outcome of this interaction. Now, this indicates that the first pairing up is happening in the middle of the T cell zone, but remember it's right at the border over here. Eventually, some of those tangoing T and B cells will migrate towards the medulla region of the lymph node, which is kind of the central portion near the efferent lymphatics. And in the textbook, they refer to this region as the medullary cords. And here they set up what's known as a primary focus, not a primary follicle, but a primary focus of cells that are dividing and differentiating. Some of those will differentiate, B cells will differentiate into plasma cells, and they will secrete antibodies that are useful early in the immune response. You also have, as we learned, B cells that are activated by T-independent antigens that make antibodies early in the immune response. And in both cases, whether it's the early T cell dependent response or the T-independent response, the early antibodies are mostly IgM isotype. You don't have a lot of class switching, and as well, you don't have a lot of somatic hypermutation for plasma cells that arise from these primary focus interactions in the medullary cords. But the other process that happens is that some of the cells migrate towards the follicles and take up residence in the follicles and begin to form what's known as a germinal center. In the textbook, they refer to this as the secondary focus, the primary focus being here in the medullary cords, the secondary focus being here in the follicle regions, leading to the production of a germinal center. And you can see there's a lot of B cells here, and there's some T cells as well dividing. And that leads to the enlargement of lymph nodes during an adaptive immune response. And you're all familiar with enlarged lymph nodes. When you go to the doctor and you have a sore throat, they feel for the enlarged lymph nodes, or if you have an infection in your arm, they feel for them under your armpit. The reason your lymph nodes enlarge is because this massive clonal expansion of T cells and B cells, in particular the B cells here in the germinal centers. So you'll be happy to know that I've decided to simplify the discussion of the germinal centers this year. There's a lot of complexity to the germinal center. You can see shown schematically over here, lots of cell types, lots of long names. Basically what I want you to know is that there's a specific architecture to the germinal center. It includes a lot of activated proliferating B cells, but it also includes dying B cells. And those can be eaten up by macrophages that are abundant here, which I don't think are shown in this, but are mentioned in the text. So B cells are actively proliferating, but many are also dying, and they're being rapidly taken up by macrophages to clear the area. But there's also T helper cells, shown here as TH, and another type of cell which is really important to know, which is the follicular dendritic cell, or FDC. And that's shown here, FDCs. So let's not talk about these two real images of germinal centers. Just remember that germinal centers are areas within the follicles that develop an organized structure with activated B cells, helper T cells, follicular dendritic cells, and then macrophages that are eating up the dying B cells. Well, why are the B cells dying? Because some of them are not winning the competition for antigen after somatic hypermutation. Now, the competition for antigen is largely mediated by these specialized cells known as follicular dendritic cells. But basically what they do is they trap immune complexes that have formed during the early stages of the immune response. Complement that has coated the pathogen or pathogen fragments during its initial encounter in the localized tissue or in the lymph node itself. So we won't go through the details here, except to say that antigens, in this case a viral antigen, it could be bacterial, get coated with complement fragments. So you all remember C3b. And eventually they get degraded to a form that can be recognized by complement receptors, CR1 and CR2, that are on the surface of FDCs. So I think we heard about complement receptor 1 when we talked about red blood cells clearing immune complexes. We heard about CR2 as part of the co-receptor on Bcells, but that's not the only cells that they're found on. They're also very abundant on these long tentacles or dendrites of the follicular dendritic cell. And that allows them to trap these immune complexes. They also have Fc receptors that allow them to trap any antigens that have IgG coding. So I want to say a few things about follicular dendritic cells. First, you can see that in an electron micrograph, they do have this tremendous octopus-like shape with these long dendrites, but they are not related to classical dendritic cells. They're not even produced by the hematopoietic system. They don't come from a hematopoietic stem cell. They're called dendritic cells because of their morphology, not because of their relationship to classical dendritic cells that present antigen with class 1 and class 2 MHC. And also to remember that because they have all these complement receptors and Fc receptors and trap antigens, they can store and trap those antigens and serve as a long-lasting depot or source of antigen for display to B cells in the germinal centers. So it's really like a warehouse or a depot displaying any antigens that are around. So B cells can test their somatically mutated heavy light chains for affinity. So figure 915. Let's see how that works. An activated B cell forming a cognate interaction with a helper T cell. We've learned about all the molecules here, the cytokines that are produced. That induces class switching, but it also induces somatic hypermutation. Now, a couple of things can happen. You can have no change in affinity or a low affinity. Forget about this term centrocyte. It's just a germinal center B cell. If it has a low affinity surface immunoglobulin, it doesn't bind to the antigen, it doesn't get an activation signal, and it can't interact further with helper T cells. So the B cell receptor is not cross-linked, and it cannot present antigen to T cells, so it dies by apoptosis. So I probably should tell you a centrocyte is just another term for a germinal center B cell. Okay? Now, if you have a B cell undergoing somatic hypermutation that leads to an increase in affinity, so high affinity surface Ig, it can recognize the antigen on the surface of the FDCs. That can lead to BCR cross-linking and antigen presentation. And the initial signal through that BCR, as well as the additional help it gets from this helper T cell, allows it to survive and divide and complete its differentiation into either a plasma cell or a memory B cell. So here's a plasma cell. It's just a striking image because it shows you a plasma cell is basically a nucleus and then a large cytoplasm that's entirely devoted to producing and secreting antibodies. So this is all endoplasmic reticulum, Golgi, and secretory vesicles. That's what it is. It's a factory for producing antibodies. And if you remember from the dreaded monoclonal antibody lecture, myeloma cells are tumors that form from plasma cells but retain the ability to secrete antibodies. The myeloma cells that are used that most patients have are secreting antibodies, and that's one of the reasons that they cause problems is because you have all this Ig in the blood. But the ones that are used for fusions for hybridomas are mutant myelomas that can no longer make their own antibodies but are able to secrete antibodies after fusion with a real B cell. So the other fate is the memory B cell, and there'll be a lecture or part of a lecture talking about immunological memory of T and B cells later on in the course. Okay, so any questions about the germinal center reaction, as it's called? Follicular dendritic cells? It's worth thinking about this process and why it leads to affinity maturation. The somatic hypermutation is random. It's not entirely random because it's focused on the V regions, but the cell isn't trying to produce a higher or lower affinity B cell receptor. It doesn't know what the antigen is. It just produces in these different clones, different mutations, and then there's this Darwinian selection for those with higher affinity. They survive because they can encounter the antigen, bind it, present it to T cells, and get the survival signal. Those that lose that competition are destined to die by apoptosis. So what you end up with is higher affinity over the course of the immune response. Okay, so we talked about somatic hypermutation and affinity maturation as a consequence of the germinal center reaction. Now we'll come back to the isotype switching. I told you before the midterm that isotype switching was different than VDJ recombination because it's not random. It's directed by cytokines produced by T cells. Now we're going to return in figure 916 to talk about what those cytokines are and a little bit about what those different isotypes are that are induced. So this is a complicated table. All I really want you to know that IL-4, which we introduced as important for B cell activation, also is a strong driver of IgG1 and IgE. And IgG1, we've learned, is good at activating complement. It's good at neutralization. Good at binding to a lot of Fc receptors, so opsonization. IgE is the key isotype that binds to mast cells and induces allergy. So if you have too much IL-4 or you tend to make IgE antibodies, that can predispose you to allergy. And this is a hallmark of a type of T cell, helper T cell known as a Th2 cell. This is one fact that is a little jumping the gun that you will learn about in chapter 8. It's in your notes now. You can come back and review it later. Interferon gamma is the hallmark of another subset, the Th1, which we did touch on earlier in the course. This inhibits those outcomes while increasing the production of a few other isotypes, in particular IgG2A and IgG3. You don't need to memorize the rest of this. It's interesting. You might see that TGF-beta induces IgA. It turns out TGF-beta is rather abundant in the mucosal immune system in areas like the lamina propria, which tends to induce plasma cell differentiation to IgA, which is the isotype you need for transcytosis and neutralization of toxins and bacteria in the gut and other mucosal surfaces. So just also want to remind you that IgM is produced early in the immune response from the TI, T-independent antigens, and from the early T cell dependent antigens. But as the response continues and in the germinal center, most of the other cytokines that are produced will suppress the expression of IgM and induce switching to other isotypes. And then the last thing I want to say for this lecture is returning to a little bit of clinical immunology. We learned about the enzyme AID, which is required for class switching and somatic hypermutation through its action on as a deaminase of cytosine residues. So if you can't do class switching, you can't make IgM. If you can't do somatic hypermutation, you can't have high affinity. There's another form of hyper-IgM syndrome that's not caused by AID. And that is caused by the absence of CD40L, the protein or the gene encoding the protein expressed on activated T cells that's required for that initial stage of B cell activation during a T cell B cell cognate interaction. So here's a lymph node from a patient with hyper-IgM syndrome. There are no germinal centers in response to a vaccine, let's say, or infection. And in a normal lymph node, you would see these germinal centers that stain very clearly through this particular histology. So patients who have hyper-IgM, they have immune responses, but they can't ever switch, and so they tend to make more IgM. They don't make IgG or IgA, and because they don't have IgE, they also don't make good allergic responses. Now, CD40L is encoded on the X chromosome, so this is a disease that mainly affects boys, whereas AID is autosomal recessive. Any questions about this last bit about T cell cytokines directing isotypes and CD40 ligand required for all of these. Okay, so just two more things. Just if you came in late, your exams are on the right, free candy in the middle, drop off your regrades on the left. Really enjoyed teaching you guys. It's been a good group, good questions, good office hours. I will be back for the review session before the final, and I'll have a office hour. My last office hour will be next Monday. But otherwise, this is the end for me, and look forward to seeing you at the final exam. Thank you.